Good day, this is the Professor, CBMD PhD, and today's Wednesday, November 1st, 2023. It is 12:41 and 27 seconds, AM Eastern Standard Time, and today's research, psoriasis and cancer. So let's hop to it here. The scientific investigators and microbiologists say psoriasis, not caused by spontaneous mutations in skin cells, date October 26, 2023. Source: Wellcome Trust, Sanger Institute. The genetic analysis of psoriasis shows somatic mutations are not linked to the development or the spread of the condition. The scientific researchers say sequenced skin samples from 111 people with psoriasis. They didn't find any mutated genes in the psoriatic patches that weren't also mutated in the individual's unaffected skin tissue. The microbiologist suggests that unlike other inflammatory diseases, such as inflammatory bowel disease or chronic liver disease, somatic mutations were not responsible for the start or spread of psoriasis. And confirming that psoriasis is not caused by any somatic mutations enables researchers to continue to explore other avenues. Over time, all cells in our bodies will accumulate mutations known as somatic mutations. These can arise from replication errors, chemicals, or environmental factors. While some of these mutations can lead to cancer, many are harmless when a mutation gives the cell an advantage over its neighbors. It is known as driver mutation, and this allows the mutated cells to grow and spread. Recently, research has begun to explore the possibility of driver mutations causing non-cancerous diseases by impacting the function of the tissue or influencing the spread of disease through the body. In previous work, these mutations have been shown to have an impact on diseases, such as inflammatory bowel disease. In this new study, researchers explored if the same was true for psoriasis. As psoriasis is a chronic inflammatory, immune-mediated disease that causes patches of the skin to become flaky or sore. The current cause of the condition is unknown, and it is estimated that 125 million people worldwide, 2 to 3% of the total population, have psoriasis. The team took samples from the forearms of 111 people with psoriasis, taking samples from psoriasis patches and healthy skin. They used laser capture microdissection to isolate 1,182 samples, which were then analyzed by whole genome or exome sequencing. They found minimal differences in the types of mutations seen in healthy skin versus psoriasis patches, and only a slight increase in the number of mutations. In addition to this, no functional differences were seen between psoriasis and non-psoriasis tissue, suggesting that the condition is not linked to a specific somatic mutation in the skin. The team identified 4 new driver mutations that gave skin cells an advantage over their neighbors, all found in both psoriasis, patches, and other skin tissue. They also found a mutational signature linked with the use of Zorolens, a compound sometimes used as part of a treatment for psoriasis flare-ups. However, these mutations were found in patients who had been prescribed Zorolens, along with those who hadn't, suggesting that it could have come from environmental exposure. And by studying somatic mutations in non-cancerous conditions has only become possible recently thanks to technological advancements. The genetic analysis of non-cancerous diseases can help identify new driver mutations such as those they describe. This adds to their growing collective knowledge about the impact of mutations on cancer and other diseases, as well as showing that certain treatments can influence the mutational landscape of a tissue. As psoriasis is a condition that affects millions of people around the world, impacting their quality of life, and very little is known about why it happens and how we can treat it. While their research did not find a gene where somatic mutations increase susceptibility to psoriasis, they are able to quantify the mutational consequences of Zorolens exposure on the skin, defining a mutational signature that may help future research. They also found that the way in which skin cells develop from stem cells is, reassuringly, unaltered by psoriasis. The replacement of non-mutated cells with those containing an advantage can have an impact on the functioning of the tissue, potentially contributing to common complex disease, risk or influencing the disease progression or response to treatment. As previous work found, mutations that could be linked to the development of high BD, exome sequencing targets, and sequences coding regions of the genome referred to as the exome whole genome sequencing captures and sequences the entire genome. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.